Hi everyone, good morning. Hi everyone, very good morning. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. So happy morning everyone. How are you doing? Hi Suhashini, hello Pavish. Good morning, good morning and good morning. And today we are again going to continue principles of inheritance and variation. Kids, this chapter is going to take time, right? I'm not going to rush. We will go slow and we will understand each and every concept, and moreover, in every lecture there will be the NCERT reading as well. Fine? So you are going to get best out of these sessions. All you need to do is you need to be consistent, and please, while watching the lecture, make sure that you are making the notes and your NCERT should be with you. Okay? Yeah, bache, whenever you want to join Sumati, you can, but again, I will say that join on time so that, you do not miss more lectures. Good morning. Thank you, Sumati. Thank you so much, guys. I hope you all are finding these videos useful. I hope you all are utilizing this content in a good way. You are revising. If you will revise, if you are consistent and you are solving MCQs, trust me, then there is nothing to worry about. For sure, you will get 360 out of 360. Okay. So I was checking the videos, 2 days back. 2 days back, I gave you one homework to write a note on phenocopy. I'm very happy to see your comments, guys. Very good, very nice, right? Even let me tell you one thing, that I was not even aware of these examples. Okay, so thank you so much. Thank you so much. Because of you, even I got to know something new. So here you guys can see, very good, bache, Medico, Suhashini, Ishfa, Akanksha, and Sahithi, Priyanka, Trisha, Karthi, Sneha, Chandan, and sure, bache Janak, I will start plant physiology. So very good, very good. See, you all have given the examples here, right? I'm very happy to read that, and very nice that you people are studying in such a good way. Excellent, excellent. And start solving the MCQs as well, okay? Please start solving the MCQs as well. Fine. Every day, it should be your habit that from physics, chemistry and biology, minimum 20 questions you should solve every day. Minimum 20, right? Maximum can be 50 or 100. It depends upon you. But for all the 3 subjects, 20 should be fixed. Just as, let's say, today I was studying physics and one chapter was very interesting and I need to spend more time on that particular chapter. What will I do? I will solve 20 questions of biology. I will solve 20 questions of chemistry, and for the physics, right, I will give extra time, and I will solve 50 to 60 questions. But I am not going to miss other subjects as well, right? And I know that today I need to pay more attention on physics, so obviously I'm going to do that. Clear? Clear? So that's what you need to understand. You have to balance all the 3 subjects together. Fine, bache. And for your best preparation, what we are providing you. We are providing you your Avengers 2.0 batch, and right, bache, here in the description box of any of the video that we teachers put, you can see in the description box, there will be the batch link as well, and there will be the Telegram channel link for our official Telegram group as well. Clear, bache? Clear? So this is what you have to do. Just click on this batch link and add your number, your email ID, join this batch as soon as possible. So currently in Avengers 2.0, we are discussing human physiology, and we are talking about the body fluid and circulation chapter. Okay, so soon it will be excretory, then locomotion, and then comes your neural and the chemical control and coordination. And trust me, you are going to love that flow of the sessions. Okay, not just for the biology, for the physics and chemistry as well. So want to see you all soon in this batch. Okay, so let's start the topic today. Let's start the principles of inheritance and variation, and let's start with the quick revision. So all of you, just let me know in the chat section, the characters that Mendel studied during his hybridization experiment. Everyone just answer the question quickly and then we'll proceed. Yes, everyone just answer the question quickly. Name the characters that Mendel has studied in his historical experiment on pea plant, which he conducted for 7 years. Okay, which he conducted for 7 years. Tell me. Name of the characters, everyone. Quickly type it and hit the like button. Share the video link with others. And let me tell you, bache, it will hardly take 10 seconds for you. After the lecture, please always comment. Always comment that whether you like the video or not. That's a request. I know 7 characters were there, but you have to name that 7 characters. You have to name that 7 characters. Quick. Yes. Let's start. So you know that stem height, seed shape, seed color, flower color, flower position, pod shape, and pod color. Clear, bache? So these were the 7 characters and they are in pairs. So basically total 14 traits were there. Okay, total 14 traits were there. That's what we have studied in the last class. And in the last class, bache, we were talking about some postulates, right? We have discussed some postulates, right, bache? And even in the last class, we have discussed inheritance of one gene, right? And moreover, inheritance of, yes, we have studied inheritance of one gene and inheritance of, yes, 2 genes as well. So basically we have studied the monohybrid cross and the dihybrid cross in the last class. Okay, that's what we have studied. So now when you know that, so Mendel has studied the inheritance of one gene as well and 2 genes as well, right? And he gave some postulate number 1, which used to say that when it comes to the factors, they occur in a pair, right? That unit factors, they occur together in the form of a pair, right? And they occur together in the form of a pair, right, bache? Right? And these unit factors, they are discrete units. They are not going to show the blending. They are not going to show the blending, the mixing. That's what we have discussed. And secondly, if you have in a pair of factors or simply in the pair of that alleles, if you have 2 different alleles, then the only one is going to express itself. Right? If you have 2 different alleles, let's say one is capital T, another is small t. So here they are talking about the heterozygous condition. And you know heterozygous is always dominant. Why? Because we have the dominant allele here and it is going to express itself. Clear, bache? The third postulate was saying that when you talk about this pair of alleles, right, during gamete formation, they will get separated. During gamete formation, they will get separated and each gamete is going to receive only one allele for a particular character, right? Each gamete will receive only one allele for a particular character. So we used to say that gametes are pure. They have just one allele for a character. Is that clear? Is that clear? So on the basis of that, what we explain the law of segregation, the law of purity of gametes, right, which is universally accepted. Okay, today we will discuss about the codominance, about the incomplete dominance. So there you will see that law of dominance is not applicable. So we used to say that law of dominance is not universally accepted, but law of segregation is, it is the law of purity of gamete as well. Then bache, we talked about the 4th postulate where we have studied the inheritance of 2 genes. That's what we have discussed. We discussed the inheritance of 2 genes, right? So here we talked about the dihybrid cross. Remember? We talked about the dihybrid cross. I hope you remember. And you know that we used to make Punnett square given by your R.C. Punnett, isn't it? Today we will read it from NCERT as well. We will mark it in NCERT as well. So Punnett square, right? We use Punnett square to write all that conditions and to calculate all that probability. So that is given by Mr. R.C. Punnett. Okay, by R.C. Punnett. So we studied the dihybrid cross in the last class, right, bache, where the phenotypic ratio is 9:3:3:1. What is the phenotypic ratio? Which ratio it is? It is the phenotypic ratio. Right? So it is 9:3:3:1. That's the phenotypic ratio. Clear? 1 point. And now what's the second point here? See, from this dihybrid cross, what we studied the law of independent assortment, right? This is the quick revision of what we have studied yesterday. Law of independent assortment. What is it? Law of independent assortment. And now this law is also not universally acceptable. Whenever you will talk about the linkage, the law of independent assortment is not applicable. Okay, whenever we talk about the linkage, the law of independent assortment is not applicable. Now, as per this law, when we talk about the inheritance of two different genes, okay, then the segregation of the alleles of that gene, the segregation of the alleles of that genes, they are not affecting the segregation of each other. Like if you am taking round seed shape and the yellow seed color, right? If I'm taking round seed shape and yellow seed color, if I'm taking this character, then the segregation of the alleles of shape, they are not going to affect the segregation of alleles of that color, right? R is not affecting the segregation of the Y. And during gamete formation, right? During gamete formation, okay, so R can, like they are independently they are segregating from each other, and randomly they are meeting in the next generation. Like if I talk about the gamete formation here, remember this part, bache? Remember this part, F1 hybrid? Yes. So Hashini, do you remember F1 hybrid? So we have to do the selfing, but before that gamete formation needs to be there, and for that we use the fork method, right? So R can go with capital Y as well, with small y as well. Small r can go with capital Y as well, small y as well. So here you will get capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So these are the gametes. Clear, bache? These are the gametes. Okay, okay. So when we are going to do the selfing of this F1 generation, okay, we will get the ratio 9:3:3:1. Fine. This is the phenotypic ratio. Okay. And today I will tell you that how to calculate the genotypic ratio as well. Done? How to calculate the genotypic ratio as well. So just please make a, okay. So make a table. Okay. So now you can even go for the trihybrid cross. This is how you have to make the gametes. You can even go for the you can go for any of the cross, right? We will in coming lectures we will also talk about the polygenic inheritance. There you will see the involvement of many genes as well. Okay, so now how can you calculate the number of gametes? Like here when I was talking about the dihybrid cross, I told you that each parent is going to form, when it is a heterozygous dominant, each parent is going to form 4 different types of gametes, isn't it? That's what I said, Suraj, right? But how can we calculate that? This is what you need to understand. Okay, this is what you need to understand. So let's say when it is your, yeah, so first of all what you need to understand, the crosses. Okay, the crosses. Just say, let's say when the character, the number of traits that you are considering, if it is 1, if it is 2, if it is 3, like one trait means you are talking about like monohybrid cross. Am I right? That in a particular experiment, when you are considering only one character or one trait, means you are talking about the monohybrid cross. Yes or no? You are talking about the monohybrid cross. Yes or no? Yes or no? And if you are taking 2 traits, 2 characters, dihybrid cross, and if it is 3, that in that experiment, what are you doing? You're going for the trihybrid cross, yes or no? You're going for the trihybrid cross, yes or no? Now, how to calculate the number of gametes here, right? How to calculate the number of gametes? For calculating number of gametes, there is a formula, that is 2 raised to power n. What is that formula? 2 raised to power n. N is the number of character or number of trait here. Okay, so here it is going to be 1. So 1. In monohybrid cross, number of gamete 1. Here it is n, right? N is 2. 2 characters are there. So it is going to be 4. Here it is 3, right? 3 characters, or you can say that 3, right? 3 characters are there. So here it will be 8. 8. Now just look at it. When it is the dihybrid cross, that's how you are going to make the gamete. That's how you are going to make the gamete. Yes or no? That is how you are going to make the gamete. Yes or no? It is 2 basically. By mistake. 2 raised to power 1 means 2 only. 2 raised to power 2 means 2 into 2, 4. 2 raised to power 3 means 2 into 2. Like 8 it is. So here you guys can see number of gametes are 4. And when you were talking about the monohybrid cross, capital T, small t, okay, then it was One gamete is capital T, another is small t. Number of gametes, they are 2. Here number of gametes, they are 4. And same way, if you will take the example of a tri-hybrid cross, okay, same way, if you take the example of a tri-hybrid cross, then what is going to happen, bache? What is going to happen here? Here you are going to see, yes, here you are going to see 8 gametes. What will you see? 8 gametes will be there, right? 8 gametes will be there, okay? Okay, so that's how, right? Right, that's how you need to figure it out. Okay, done? Yes or no? Yes or no? So I'll tell you about that cross. We will make it and we will discuss it as well. Fine, we will discuss it as well. So here, right, so this is about the number of gametes or number of gametes also, and right, this is about the number of gametes. Now when you need to calculate, yes, when you need to calculate number of zygotes, next is what? Next is what? Next is we need to calculate number of zygote. What we need to do next? We need to calculate number of zygotes. So here it will be gametes raised to power n. Okay, here it will be gametes raised to power n. 2 raised to power n, 4 raised to power n, 8 raised to power n. So 2 raised to power n means, right, n is the number of gametes, it is 2. Okay. And here it is 16, right? Here it is 16 and here it is, okay, so here basically when you are calculating number of gametes, what you have to do in the case of number of gametes, right, in the case of number of zygotes or gametes, so number of zygotes divided by offsprings it will be. So actually it will go like this, like you have to consider n as a 2. Yes, here you have to consider n as a 2 basically. N will be 2 here and you have to consider the number of gametes. Fine, number of gametes then, right? So it will be 2 into 2, that's 4. 4 into 4, that's 16. 8 into 8, that is going to be 64. Okay, that is going to be 64. And here we have one more formula, we'll let you know about it. Okay, so here number of zygote, if you want to calculate number of zygote, so this is what you need to do. Okay, this is what you need to do. Clear, bache? So number of zygotes or offsprings is gamete raised to the power 2, right? Gametes raised to the power 2. So this n is always 2 here. Okay, this n is always 2 here. That's how you have to calculate. Fine, that's how you have to calculate. Fine, done, bache? And this number of zygote, na, it is also dependent on the ratio part. We'll let you know about it. So gametes raised to the power 2. So you can also write gametes raised to the power n. N is the 2 here. Is that clear? Is that clear? Okay, so next is the number of phenotype and number of genotype. What we need to calculate next? It is the number of phenotypic ratio and the number of genotypic ratio. Number of phenotypic ratio and the number of phenotypic ratio and number of genotypic ratio. See, you know, no doubt we are going to talk it in this, we are going to discuss it in this way, that we have monohybrid cross, dihybrid cross, and then we have trihybrid cross. But do you know how do I remember it? How do I remember it, this particular part? Right, see, to remember the ratio of monohybrid cross is going to be very easy for us, right? When it is phenotypic ratio, it's 3:1. When it is genotypic ratio, it's 1:2:1, right? That's what we have. When it is the monohybrid cross, it is 3:1, and when it is the genotypic ratio, it is 1:2:1. So in the case of monohybrid cross, fine, in the case of monohybrid cross, when you need to figure out the phenotypic ratio, see, Two ratios you got now. So here the formula is 2 raised to power n. That's how I remember. Here we have 3 ratios. So 3 raised to power n will be the formula. That's my way to remember it. Okay. Monohybrid cross is having the ratio 3:1. 2 ratios are there. So 2 raised to power n if you want to calculate the phenotypic ratio. And when you talk about the genotypic ratio of monohybrid cross, it's 1:2:1 means 3. Right? It's 3 ratios are there now. So 3 raised to power n. That's how I remember. Clear? That's how I remember. So see, 2 raised to power n means 2 only. 2 ratios you are going to get, and you know that that's 3:1. Now apply it on dihybrid cross. The same formula. 2 raised to power n. N is what? N is 2 here. N is 2 here. So 2 into 2 will be 4, and you know the ratio here. It's 9:3:3:1. Okay, it's 9:3:3:1. Now when it is trihybrid cross, the ratio is going to be 2 raised to power n. N is 3 here. So it is 2 into 2 into 2, 8. So 8 ratios will be there. We will solve it now. Okay, we will solve it now. Now come to the genotypic ratio. So it will be 3 raised to power n. N is 1 here. So it will be 3. And you know that in monohybrid cross, it's 1:2:1. And here it will be 3 raised to power 3. Here it will be 3 raised to power 3. Here it will be 3 raised to power 2. 3 3s are 9. We'll tell you right now. And here it will be 27. Right? So this is the number of ratios. Now I'll give you one example. But do you know about the product rule? Do you know about the product rule? Yes. Do you know about the product rule? Tell me quickly. Everyone, do you know about the product rule? Yes. I'm waiting for your answer. Be quick. Do you know about the product rule? If yes, then good. Otherwise, let me tell you. See, I want to calculate the phenotypic ratio for the dihybrid cross. Okay. Just assume that you are not aware of the ratio of dihybrid cross. Just assume. Okay. Just assume that you don't know the ratio of dihybrid cross. I can apply the product rule to calculate the genotypic ratio of dihybrid cross, of trihybrid cross, and if there is any tetrahybrid cross, now I can check that ratio with the help of this product rule. What is this product rule? Now let's say I need to check the phenotypic ratio of dihybrid cross. And I just know the ratio of monohybrid cross. So I will apply product rule. So what product rule is saying? 3:1 because it is a dihybrid cross. So I will multiply. 3:1 is the phenotypic ratio of monohybrid cross. Right? So I am talking about a dihybrid cross. Di means here you are taking 2 characters. So you are going to multiply these ratios. Okay? You are going to multiply these ratios. 3 3s are 9, 3 1s are 3, 3 1s are 3, and 1. You got your ratio. You can check the calculation. Right? You can check the calculation. And one more example. Same way, if you want to calculate trihybrid cross ratio, then 3:1, 3:1, 3:1. That's what you have to do. That's what you have to do. So you already know that 3:1, 3:1 is 9:3:3:1. Then multiply it with this. Multiply it with this. Right? You will get your ratio. Wait, see this. 9:3:3:1. Multiply it with this. So that's how you will go. See, genetics is interesting, but it's very, very, very interesting. Okay, so 3 9s are 27. But check this. Is it the phenotypic ratio for the trihybrid cross? You are going to check it. Go for it quickly. Is it the phenotypic ratio for a trihybrid cross? Is it the phenotypic ratio for a trihybrid cross? Just check it. When you are hybridizing 2 heterozygous dominant. Just check it. Right? You can just confirm it. Okay, you can just confirm it. So this is your phenotypic ratio. This is your phenotypic ratio. Right? This is your phenotypic ratio, yes or no? Okay. See, 8 is written now. So 1, 2, 3, 4, 5, 6, 7, 8. Look at it. 2, 3 is to 1. 4, 9 is to 3 is to 3 is to 1. Now it is 8. So this is the ratio. This is the ratio. So it is not mandatory that you have to cram it. If you know the product rule, you can calculate any of the ratio. So let's say in the NEET examination, they're asking you the question, right? They're asking you the question, they'll give you the heterozygous, right? They will give you the 2 heterozygous dominant, and then they are asking for the ratio. There is no need to make the cross. There is no need to make the Punnett square. There is no need to calculate the ratio then. You can simply apply the product rule if your basics are clear. Okay, and same we can use for the genotypic ratio as well. Okay, same we can also use for the genotypic ratio as well. So now you know that genotypic ratio of a monohybrid cross is 1:2:1. Yes, genotypic ratio for a monohybrid cross is it's 1:2:1. Now if you want to calculate the genotypic ratio for a dihybrid cross, what will you do? 1:2:1 multiply 1:2:1. So what will you get here? So 1, see, this is the ratio. This is the genotypic ratio. Okay, this is the genotypic ratio. So see, 1, 2, 3, 4, 5, 6, 7, 8, 9. And see here, 9. So tell me, any doubt? Any doubt? So it's simple maths. It's simple maths. Let me show you it from NCERT. Dihybrid cross. Okay, this is your dihybrid cross. This is your dihybrid cross. So you know that the ratio is, phenotypic ratio is 9:3:1. So see, round yellow, 1, 2, 3, 4, 5, 6, 7, right, 8, and this 9. They are round yellow. They are round yellow. 3 are your round green. Next 3 are your wrinkled and yellow. And next one is your wrinkled and green. This is the phenotype. This is the phenotype. Now just look at the genotype. See, look at the genotype. This one is homozygous dominant, isn't it? Homozygous dominant. Yes or no? This one is homozygous dominant. Yes or no? Yes or no? Quickly. And look at this one, heterozygous dominant. Okay, and heterozygous dominant. Fine. And heterozygous dominant. Clear bache? Clear bache? And when you look at this, round color is homozygous dominant, yellow color is heterozygous. So in this way you can check the genotypic ratio. In this way you can check the genotypic ratio. So you have how many different types of individuals you have on the basis of that. Just check your chart. It is going to be dihybrid. Okay, it is going to be 9, right? 9. And this is the ratio, 1:2:4:2:1:2:1:1. So that's how you have to check. Got it? Got it? So let's quickly revise it and then we will start your test cross. So these are the 7 characters. And inheritance of 1 gene we have studied. We have studied the inheritance of 2 genes. Now we know about the laws as well. We know about the definitions as well. Okay? Okay. So that's how you have to make the gametes. Fine. That's how you have to make the gametes. Clear? So when you want to check the number of gametes for a monohybrid cross, the formula is 2 raised to power n. N is what? Number of characters here. right? So on the basis of that, you checked it. When you want to check the number of zygote, so gametes raised to power 2. It is gametes raised to power 2. So gamete is 2 here, 2 raised to power 2, 4 raised to power 2, 8 raised to power 2. Clear, bache? Same way you can check for the tetrahybrid cross as well. Next is number of phenotypic ratio, 2:n. So 3 is to 1, this and this. You can check it. Then when it is the genotypic ratio, 3 raised to power n is the formula. And after applying the product rule, you can calculate the ratio. Clear? You can calculate the ratio. Any doubt? Yes, any doubt? Sure? Yes, bache, sure? Okay? Okay? So next is the test cross. After discussing the test cross, na bache, see, in the NCERT it is given now. So after discussing the test cross, we will start the NCERT reading. Okay? We have to discuss the test cross first and then we will start the NCERT reading. So tell me, what do you know about the test cross? What do you know about the test cross? Tell me quickly. What do you know about the test cross? Yes. So how many of you are doing this rule for the first time? How many of you are doing this rule for the first time? Yes, quickly tell me, how many of you are doing this rule for the first time? It's a very simple rule for genetics, bache. Okay? It's a very simple rule. And if you understand these concepts, now you can save your time in the NEET examination. These days, they are not asking, so many questions from the genetics, so many numerical based questions from the genetics, but you should prepare yourself. Okay? You should prepare yourself. So now the topic that we need to start is, it is the back cross actually. What we are going to start, it is the back cross actually. What is the meaning of a back cross? Back cross means, right, the F1 individuals, okay, F1 hybrids, we are going to cross them with their parents, right? The meaning is back cross. Okay, so in which F1 individuals are crossed with any of their parent, right? F1 individuals are crossed with any of their parent. Clear? With any of their parent. It is the back cross. Like for an example, simple. If it is capital T, small t, you are getting capital T, small t. Now if you are, this is the F1 individual. If you are crossing it with any of the parent, it can be capital T, it can be small t, then you are going to call it as back cross, right? You are going to call it as back cross. Now when you talk about this back cross, it is having two types. Okay, it is further having two types. One is the out cross and another one is the test cross. One is the out cross and another one is the test cross. And in our book, in our NCERT, the test cross is given. Clear, bache? In our NCERT, what is given? In our NCERT, your test cross is given. Now, what is this out cross? See, when you talk about the out cross, here, F1 individual, we are going to hybridize, or you can simply say cross F1 individual with dominant parent. Okay? With dominant parent. Right? Individual will be hybridized. Yes, individual will be hybridized with the dominant parent. Clear, bache? Clear, bache? So this is your out cross. This is your out cross. Now, what we need to understand? We need to understand the test cross. And this test cross is used to determine the unknown genotype. We can use this test cross to determine unknown genotype. Focus on this word. It is genotype, not the phenotype. Okay, not the phenotype. So what is going to happen here, bache? In test cross, F1 individual. Here cross your F1 individual with homozygous recessive parent. With homozygous recessive parent. Are you getting it? Are you getting it? Clear, bache? So you have back cross. So F1 individuals are, right? F1 individuals are crossed with any of their parent. Clear? So back cross is having two types. Back cross is having two types. One is your out cross, another is your test cross. And when you are talking about the out cross, here you are going to cross F1 individual with the dominant parent. Here you are going to cross F1 individual with the homozygous recessive parent. Now just focus on this part. It is homozygous recessive parent. And why are we mentioning, why are we specifying homozygous recessive parent? Because bache, recessive trait can only express itself in the homozygous condition. Okay, recessive trait can only express itself in the homozygous condition. That's why we are saying it is homozygous recessive parent. Clear, bache? So for an example, and moreover what I just said, that here you have to check the unknown genotype. Right, here what you need to check, bache, Priyanka, Ishwat, you need to check the unknown genotype. Now what is the meaning of it? Let me quote one example here. Fine. Let's say you are looking at a plant. Fine. You are looking at a plant. By looking at it, you can say now that it is tall or not. Or if you are looking at a flower color, by just looking at a flower color, you can say, okay, it is red in color, it is blue in color, it is yellow in color, right? So phenotype, you can see it clearly with your eyes. It is the external appearance, right? It is the morphological appearance. It is the external appearance. Yes or no? Yes or no? It is the morphological appearance. It is the external appearance. You can just look at it and you can just tell it. Yes or no? Yes, Satyam, Priyanka, Ishwat, Nandini. Is it so? Of course. So now if I'm looking at this plant and I'm saying that this plant is tall, so there are two possibilities. Okay. Okay. If it is dwarf, then it is understood that dwarfness means it is a recessive trait. So it will express itself in homozygous conditions. है ना? It will express itself only in homozygous recessive condition. Right. But I just need to check whether it is homozygous dominant or the heterozygous dominant. Okay. I'm just looking at this plant and I'm saying that that plant is tall. So I just need to check what is it? Is it homozygous? Is it homozygous tall or heterozygous tall? Is it homozygous tall and heterozygous tall? Yes. So it means two possibilities are there. Either it can be capital T, just a minute. Either it can be capital T, or it can be capital T, small t. Either it can be capital T, or it can be small t, right? So the genotype is unknown. I don't know the genotype of this plant. I want to check the genotype of this plant. Okay, I don't know the genotype of this plant. It's just that I want to check the genotype of this particular plant. So let's take two cases. Case one, when we are assuming that it is homozygous tall. Fine, it is homozygous tall. Means then your unknown genotype is this. Case 2, when I'm saying that it is heterozygous tall, then it is capital T, small t. Now I just need to confirm it. Okay, so I will go for the test cross. So if it is a test cross, I will cross this individual with homozygous recessive parent. This is, let's say this is the F1 individual. I'm crossing it with the homozygous recessive parent. Yes or no? Quickly tell me, students, yes or no? So now you know that what will be you're going to cross them. The gametes for this parent will be capital T, the gamete for this parent will be small t. Here it will be capital T, small t, and small t, small t. That's what we have. Yes, AET, that's what we have. So see, capital T, small t, capital T, small t, capital T, small t, capital T, small t. Means here, after this cross, all the individuals are showing the dominant trait, right? All the individuals are showing the dominant trait, right? The entire population is dominant. Now here, capital T, small t, capital T, small t, small t, small t, small t, small t. So here 50% of the population, right? 50% of the population is showing the dominant character and 50% of the population is showing recessive character. Fine, recessive character. 50-50 it is. 50-50 it is. So if it is 50-50 means the ratio is going to be 1:1. The ratio is going to be 1:1. It is going to be 1:1. 50% is homozygous, 50% is heterozygous tall, 50% is homozygous recessive. It is dwarf. It is dwarf. So what can we depict from this part? That if we are taking a test cross to determine the unknown genotype, right? How can we determine the unknown genotype? Because when we look at a particular plant, we can just notice the character. We can see the phenotype, right? So I want to check whether it is a homozygous or heterozygous tall. By looking at it, I can simply say it is tall, but I want to confirm whether it is homozygous or heterozygous. So I have two possibilities, right? I'll assume that it is homozygous tall, or I can say that it is heterozygous tall. I just need to hybridize it with homozygous recessive parent. So whenever it is a test cross, okay, whenever it is a test cross, the F1 individual will always be homozygous recessive. Okay, the F1 individual, Vidisha, will always be homozygous recessive. Clear, bache? F1 individual will always be homozygous recessive. Is that clear? Is that clear? So if the ratio is all are showing the dominant character, entire population is having the dominant character, definitely unknown genotype is homozygous dominant. If 50-50% ratio is there, unknown genotype is heterozygous dominant. I hope this part is clear, bache. I hope this part is clear. Yes. So this is what test cross is. And if you go for the dihybrid cross, okay, like let's say you are using same scenario for dihybrid cross. So in that case, the ratio will be 1:1, 1:1, 1:1, 1:1, like this. Okay, so it is the heterozygous dominant, it is the homozygous recessive parent, and that will be your ratio in the case of dihybrid cross. Okay, Satyam, that will be the ratio in the case of your dihybrid cross. So that's what you need to keep in your mind. Okay, okay, that's what you need to keep in your mind. Is that clear? Sure? Yes, everyone, sure? Are you sure? Done. So this is what test cross is, right? This is what test cross is. So see, this example is given in the NCERT. They have considered the flower color. So if all are violet, you know violet is a dominant trait. So unknown flower is homozygous dominant. If half of the flowers are violet and half are white, unknown flower is heterozygous. Unknown flower is heterozygous. That's what test cross is. So can we just revise it quickly from NCERT? Can we just start the NCERT reading, bache? Yes. Can we just start the NCERT reading? Open your book. These days, I'm feeling that I'm teaching the students of KG, right? Right? We are explaining something, then I am marking it on NCERT, then I'm telling you, open your book, right? Mark this point, put the finger on that line. And you know, it is a very effective way. Do you know that? It's a very effective way. Like when you study like this, you have to focus on this finger. Right? So it will move like that. It can increase your speed. It can increase your speed. Okay? Hannah, so now the students of kindergarten, please open up your books, right? Take out your pen, right? We are going to start the highlighting part now. We have to mark the important words. है ना? In the junior classes, this is what teachers used to say. Do you know the meaning of this particular word is this? Do you know the meaning of this particular word is that? Same way, I will tell you, do you know from this line MCQ can come? Do you know this question can be asked in this way? Right? Right? So it is not the NEET preparation. It is actually not the NEET preparation. Chalo, so focus here. So let us take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and the dwarf pea plant to study the inheritance of one gene. So they are simply saying that when Mendel conducted an experiment, when he studied the inheritance of one gene, he took the character tallness. Okay, height is the character that he used. So he collected the seeds produced as a result of this cross and grew them to generate plants of first hybrid generation. So obviously, right, we have to grow the seeds, we have to see the results. That's what Mendel did, right? That's what Mendel did. So the first hybrid generation is known as first filial generation or first filial progeny. It is known as first filial generation. It is known as first filial progeny. Mendel observed that all the F1 progeny plants were tall like one of its parents. Okay, see, all the F1 progeny plants were tall like one of its parent. Like out of that two parents, monohybrid cross it is now, one is homozygous tall, another is homozygous dwarf. But in the next generation, we'll see that all the plants are just like their one parent, which is homozygous tall. Okay. Okay, so he made similar observation for other pairs of trait as well. Other than height, he took some other characters also and he made the same observations. Okay, so he found that F1 individuals are always resembled either one of the parents and the trait of other parent was not seen in them, right? So this is what he observed, that whenever, in such cases, whenever he take the pure parents, right, pure lines for a particular character, F1 individual will always show the character of the one parent. Okay. So Mendel then self-pollinated the tall F1 plants and to his surprise found that in second generation, some of the offsprings were dwarf, means they were showing the character of the second parent. So character that was not seen in F1 generation, it was now expressed. So the proportion of plant that were dwarf were 1/4. You know the ratio 3:1. So obviously 1/4 will be dwarf in F2 population and 3/4 of the plants were tall. Clear? That's why we always take the ratio of F2 generation. Got it? That's why we always take the ratio. Just whenever you calculate the dihybrid cross ratio, 9:3:1 it is now. So again you are taking the ratio of F2 generation. It is not the ratio of F1 generation. Okay. And simply when you are just crossing, see, how are you getting this ratio? When you are crossing two heterozygous dominant. So this is what you need to remember. Let's say in the final NEET examination, this is the case that you got, right? That what will be the ratio? So do not try to calculate the F2 generation. Here simply just look at it. Heterozygous dominant, heterozygous dominant. The ratio will be this. The ratio will be this. Got it? Got it, bache? So tall and the dwarf traits were identical to their parent type and they did not show any blending. That is, all the offsprings were either tall or dwarf. None were of in-between height. This is what Mendel observed. See, it was Mendel observation and I gave you homework, I think in the last class also I didn't, but you didn't answer the question, bache. I told you na that when it comes to the pea plants, the character he selected, they were located on different chromosome, 1, 4, 5 or 7 I think, right? 1, 4, 5 or 7 if I'm not, yeah, 1, 4, 5 or 7, right? You can check it once. So he didn't find the traits like, he didn't find the inheritance pattern like incomplete dominance or codominance. So he was lucky as well, right? Or I won't say it was not luck, he worked so hard that, you know, destiny favored it. Okay. So here you guys can see that this is what he observed, that offsprings either they are tall or they are dwarf. No one was, you know, intermediate. This is what he observed. Okay. So after this topic, we will also discuss some deviations from the Mendelian inheritance. So similar results were obtained with other traits that he studied. Only one of the parental trait was expressed in F1 and in F2 both the traits were expressed in the proportion 3:1. Clear bache? Clear bache? In 3:1. So contrasting traits did not show any blending. So there is no mixing in F2 generation. Okay, mixing is not there, right? Mixing is not there. Either they are tall or dwarf, but it is not mixed. Mixed means that no one was like that, that let's say it was 10 centimeter tall, it was 3 centimeter dwarf. No one was 7 centimeter. Either they were 10 or 3, just like their parents. Okay, just like their parents. Now, based on these observations, Mendel proposed that something was being stably passed down, okay, which is unchanged from parents to offsprings through the gametes over successive generation, and he called it as factors. So this is what he observed. He was like that something is passed to the next generation from parents to offsprings from the gametes, and it is what? It is stable and unchanged. So MCQ can come from this part, right? I can use these lines for the statement based question and for the assertion and reason kind of questions. Okay, for the assertion and reason kind of questions also I can ask this question, right bache? Right, so these are the factors. Now we call that factors as genes, and Jonathan has given it the term gene. Clear bache? So genes are what? They are the units of inheritance. Again a famous question, right? Again a famous question. Genes are what? Genes are the units of inheritance. So they contain the information that is required to express a particular trait in an organism. So what the genes will contain? They contain the information which is required to express a particular trait. Clear bache? Clear bache? And when these genes, they code for a pair of contrasting traits, they are known as alleles or allelomorphs. They are slightly different form of the same gene. What are alleles? Slightly different form of the same genes, and they formed because of the mutation. So the language clear? Language clear, right? NTA can play on the basis of language here, right? In the principles of inheritance and variation, you will study some paragraphs where language is little complicated. So you need to understand that part as well. Clear? You need to understand that part as well. So this is the case that we have. So if we use alphabetical symbols for each gene, so capital will be used for, you know that capital will be used for the dominant, small will be used for the recessive. So see, in case of height, capital T is used for tall trait, small is used for the dwarf. Okay, so capital T and small t are the alleles of each other. They are the slightly different forms of each other. Doubt? Yes, Poonam bache, any doubt here? So hence in the plants, the pair of alleles for height would be capital T, capital T, capital T, small t, small t, small t. Clear? So Mendel proposed that in true breeding, tall or dwarf pea variety, allelic pair of genes for height, they are identical, homozygous respectively. And see, this is the genotype. Capital T, small t, capital T, capital T, small t, small t, they are the genotype, right? But tallness and dwarfness, it is the phenotype. So I hope you know the difference here. I hope you know the difference here. Yes. So what will be the phenotype? They are asking a question. What will be the phenotype if a plant is having this genotype? You know that here also it will be dominant. Here also it will be dominant. Heterozygous is always dominant. This is written here. So as Mendel found the phenotype of the F1 heterozygote TT to be exactly like of TT. So he proposed that in a pair of dissimilar factors, this is the point that we discussed yesterday, yes or no? This is the point that we discussed yesterday, yes or no? That in a pair of dissimilar factors, one dominates the other. So it is called dominant factor, and the other which is not expressing, it is recessive. It is the postulate number 2, right? Right? So capital T is dominant over small t, which is recessive. Done, bache? Done? So it is convenient and logical to use the capital and lower case of an alphabetical symbol to remember this concept of dominance and recessiveness. Fine. So alleles can be similar in the homozygous conditions, and it can be dissimilar, and they will be the heterozygotes. Clear, bache? So since TT is heterozygous, right, it is controlling one character height, it is the monohybrid cross between this, right? That's what we have studied already. Fine, bache Nandini, that's what we have studied already. Clear? So next is, from the observation that the recessive parental trait is expressed without any blending in F2 generation, right? So this can be asked as a question, that if you have a parent with one with the homozygous dominant trait, another with the homozygous recessive, so the trait of both the parents will express in which generation? In F2 generation. Okay, of both the parents will express in the F2 generation. Clear? Clear, bache? So we can infer that when the tall and the dwarf plant produce gametes by the process of meiosis, so alleles of the parental pair, they separate or segregate from each other, and only one allele is transferred to a gamete. Postulate number 3. It is explaining law of segregation of gametes, right? Law of segregation of gametes, remember? So this segregation of allele is a random process. Okay, they can ask you this thing. I can form a MCQ from this part. Okay, so segregation of allele from a pair of, right, from a pair of allele, if there is a segregation of allele, is it a random process or non-random process? So in the final paper, they can ask these kind of questions. So it is a random process. So 50% chances are there. One gamete can also receive capital T, it can also receive small t. So 50% chances are there. Clear? Clear, bache? So in this way, the gametes of the tall plants have the allele, and the gametes of the dwarf plants, they are also having the allele capital T, a small t in their gametes. So during fertilization, right, capital T from one parent through the pollen and small t from other parent through the egg are united to produce the zygote, right? So zygote will be then capital T, small t. So this is the hybrid, this is the heterozygous. Fine. So in your NCERT, please highlight this. Highlight this. Okay. In your NCERT, please highlight this. Right. So the production of gametes by the parents, the formation of zygote, right? It can be understood from a diagram called Punnett square. Right, bache? So it was developed by a British geneticist, which is Reginald C. Punnett, or simply you can say R.C. Punnett. MCQ, another important MCQ. So you can use this diagram to understand, right, the formation of zygotes, isn't it? The formation of zygote. So it's a graphical representation to calculate the probability of all the possible genotypes of offsprings in a genetic cross. So again, from here also MCQ can come, right? So it is a graphical representation which helps to calculate the probability of genotype. So do not get confused here. It is the genotype, not the phenotype. Okay, it is not the phenotype. So that's what you need to focus, right? So possible gametes are written on two sides. You know that, usually the top row and the left column. So all possible combinations are represented in boxes, which is there in the square form. So you know about it, right? You know about it. So here you guys can see this is the Punnett square and this is for the monohybrid cross. Okay, so these are the ratio. We have already discussed this part. Fine, bache. Fine. So F1 plant of genotype capital T, small t, when self-pollinated, they will produce the gamete of genotype this in equal proportion. So when fertilization takes place, pollen grains of genotype capital T have a 50% chance to pollinate eggs of genotype capital T as well as small t. So capital T can get fertilized with capital T also, small t also. I have already explained it, right? I have already explained it. Done, bache? Done. So see, this is what they have given. So now when you read this particular part, okay, so here what they have given, that from the Punnett square, it is easily seen that 1/4 of the random fertilization leads to this. Yes or no? Yes or no? Fertilization is a random process also. They are not selecting that capital T will fuse with capital T only or capital T will fuse with small t only. No, it's not like that. Clear? It's not like that. So 1/2 will form this, 1/4 will be this. So though the F1 have a genotype of capital T, small t, so basically here in this part, they are just trying to explain that ratio part. Is that clear? Is that clear? This is what they are trying to show. So see, phenotypic ratio of 3/4 tall plant, okay, and 1/4 is the is for the homozygous dwarf, and the genotypic ratio is this. Okay, the genotypic ratio is this. Done, bache? Done. So here they have written the genotypic ratio, okay, and they have given it in the form of a binomial expression. So see, the 1/4 is to 1/2 is to 1/4 ratio of this is mathematically condensable to the form of a binomial expression, and this is the binomial expression, right? This is the binomial expression. So actually they are expressing this ratio in the form of this binomial expression. Okay, okay, fine. So you can see it here. You can read this part from NCERT. That's how they are expressing it. Fine, bache, that's how they are expressing it. You know, na, binomial expression. That's all. Fine, that's all. Okay. So rest is again about the ratios only, right? So ultimately they have just expressed these ratios part only, that's how we will represent it. Bas, simple it is. Fine, I think it's the same slide. Yes, it's the same slide. So this is what you need to read from the NCERT. And here in this paragraph, they also talked about your test cross here. Fine, they also talked about the test cross, that from the preceding paragraphs it is clear that though the genotypic ratios can be calculated using mathematical probability by simply looking at the phenotype. So you can just look at the phenotype and you can calculate the ratio. You can apply these binomial expressions as well, bache, right? So it is not possible to know the genotypic composition. So for that, when a tall plant from F1 or F2, right, we cannot predict whether it is homozygous dominant or heterozygous dominant. So just to check the genotype, whether it is homozygous dominant or heterozygous dominant, what will we do, right? The test cross needs to be performed. Okay, the test cross needs to be performed. So in a typical test cross, an organism showing a dominant phenotype is crossed with the recessive parent instead of self-crossing. So progeny of such cross can be analyzed to predict the genotype, bache. So you know that. How are we going to predict? So we will check the ratio. So if it is 1:1, it is showing that unknown genotype is the heterozygous one. Clear? It is the heterozygous one. Clear, bache? Clear, bache? So this is what Mendel did. This is what Mendel studied, and then he depicted that loss. Okay, then he gave that loss. Clear? Clear? So let's read it, let's revise it. So based on his observation on monohybrid cross, Mendel proposed two general rules to consolidate his understanding, right? Principles. These are the principles and the laws of inheritance. First is law of dominance, second is law of segregation. So see, law of dominance is saying characters are controlled by discrete units called factors, right? So I was using the language, the factors. Why? Because it is given in NCERT. Factors, they occur in pairs. So in a dissimilar pair of factor, one is dominant over the another. So it is your law of dominance. Clear, bache? Clear, bache? So it is saying that in monohybrid cross in F1 generation, only one of the parental character will be expressed. Fine. So next is law of segregation. It is based on the fact that alleles do not show any blending, and both the characters are recovered as such in F2 generation. Clear? Alleles are not going to show the blending, and both the characters will be recovered as such in the F2 generation. Clear, bache? Clear? So parents contain two alleles during gamete formation. So these alleles of a pair, they segregate from each other such that gamete receives only one of the two factors. Okay? Okay? So homozygous parent, they will form similar gamete. Heterozygous parent, they will form dissimilar gametes in equal proportion. Clear, bache? So this incomplete dominance, codominance, rest we will discuss in next class. And see, inheritance of two genes is already explained to you. So that's your homework. You will read it from NCERT, right? This particular paragraph you need to read from NCERT. And we will discuss it in the next class. Okay, we are going to discuss it in the next class, and stay tuned to channel. Might be I'll take your session of this principles of inheritance and variation tomorrow. Maybe I will take it. Okay, you stay tuned to channel, and tomorrow you know now what are we going to do. Breathing and exchange of gases, one shot. Yes, medical. Okay, so tomorrow there is a one shot of breathing and exchange of gases, and today one more thing. See, today at 4 p.m. we are going to have one more class. Okay, we will practice the questions of sexual reproduction in flowering plant. So this class will be for 30-40 minutes. Okay, join this class and revise that particular chapter. We have completed it now. So human reproduction done, reproductive health done, sexual reproduction in flowering plants done. So I think in few days we will finish principles of inheritance and variation. Then molecular basis of inheritance we are going to start. So parallelly your 11th and 12th syllabus is going on. So in class 11th, your cell cycle and cell division, your biomolecules, structural organization in animals, your digestion and absorption, plant kingdom, animal kingdom, your living world, biological classification, it's all done. Okay, so see you all today in this particular session. And yes, bache, so see, and even your tomorrow your HSP sir is going to take, see, this amazing session. And yes, look at this video section here. See, just look at it. See, this is what he has posted, must watch, right? And see, check this video as well. Here I have explained the AIMS cutoff, right? You will understand the level of competition, okay? And you're going to feel motivated, so do check it. And in the comment section, now you know what you have to do. You just need to let me know that you like the session or not. That's it, that's all. So take care. See you all today at 4 p.m. and then tomorrow. Okay, so thank you so much. Bye-bye. Thank you.